Hi folks, it's Mr. Byers again. So I want to give a really quick summary of chapter 12. Um, it's titled as DNA and RNA, but really we also go into gene expression and how we can use the information in DNA, how our cells can use this information and form a protein. So we have our basic DNA helix structure. That structure, that DNA, is filled with genes, particular locations of information that code for the protein. But it's not as simple as seeing the information on the DNA and knowing what and forming a protein. We have to go through a few steps here. Now, DNA is inside the nucleus. That's where it's protected. You don't want your DNA to break down. It does not leave the nucleus under normal circumstances while the cell is doing its day-to-day functions. However, if you recall from the earlier units, the ribosomes, the factories that are out there producing the proteins, they're in the cytoplasm or attached to the rough ER outside of the nucleus. So somehow we have to get this information here in the DNA out to these ribosomes. To do that, we use strands of RNA. So the first strand that's formed from DNA is called mRNA or messenger RNA. And it is coded, and when it is transcribed from the DNA, it transcribes every section, both those sections that code for genes, exons, and those that aren't coding for the gene or aren't useful for this particular gene, introns. So this is what we call a rough draft, a pre-mRNA. It has to be edited. So we cut out the introns, splice those together with the spliceosome, and add a cap on either end. Now we have that mRNA transcript that can be sent out to the cytoplasm. Once it's edited and completed, just like how you have a rough draft, you don't submit your rough draft, or you shouldn't submit your rough draft to your instructor. You edit it, you read it first, you make some changes, then you submit that, your final draft. So this mRNA strand is like a final draft. We send it out into the cytoplasm to the ribosome, where the ribosome actually, those two subunits, a scaffolding of rRNA and proteins, folds around or forms around that mRNA and reads it. And so that mRNA transcript slides through that ribosome and it's read a codon at a time. So remember, DNA has those triplet codes, groups of 3 nucleotides. And so we process that in groups of 3. That's transcribed into groups of 3 codons. Those codons are read by the ribosome in groups of 3. And everywhere we see that particular codon, tRNA has a particular anticodon that attaches, that coincides with that codon, and they bring a particular amino acid. That amino acid is brought, we slide to the next codon. We attach that amino acid, we slide to the next one, we attach that amino acid. More and more and more and more and more until our protein is built. And so we go through that initiation, elongation, and then finally termination. We break off the protein and now it's finished. And so then it goes on to do its job. Maybe it's processed by the rough ER, maybe it's sent somewhere inside the cell, or maybe it's sent somewhere outside of the cell. But a protein is that actual structure and function of what we are, all of those substances. And so DNA is the information, mRNA is sort of the rough draft that can be read by the ribosomes. The protein is the actual formation of that trait necessarily. So just real basics here, real simple. We know that most genes are probably longer than this, but we have our double-stranded DNA in that triplet code. One side of that triplet code, one side of the DNA is going to be read by DNA polymerase, and we're going to form RNA polymerase, and we're going to form an mRNA strand. It's transcribed into an mRNA. So this triplet code is transcribed into codons, into groups of 3. So the first codon here is AUG. Now you may have to flip back a couple slides, but I have that list. Let's just go to it. Let's go to it here. You should memorize what AUG is. That's always going to be your start codon. But if we look here at this list, of all codons, because this is the same across life. Every living life form, these are the codons. This is how it works. There may be multiple codons per amino acid, but that codon, for example, UUU, that codes for phenylalanine. That does not change no matter what organism on the planet we are talking about. That's how it works. AUG is a very special codon. It's the start codon. It codes for methionine. Now, you don't have to memorize all these, obviously, but I would keep an eye on the start codon and the stop codons. So the very first codon that we would find, so it would bring the methionine, and then these final stop codons, they're at the end of that RNA. They act like a period or the breaks. They don't necessarily code, well, I shouldn't say necessarily, they don't code for an amino acid, but they tell the ribosome, hey, we're done. And so you're always going to see the start codon in the beginning and the stop codons at the end. If I were to ever ask you, besides those four, to name a particular codon based, or name a particular amino acid based on the codon, I would give you a chart. To use the chart, look, you have the first base, groups of U's here, that's the first one. Second base is in this column, third base is in this column. We just line them up. If we have, let's make up one, CCC, or CAC, let's make up that, CAC. We get out here, CAC, we go over to the C, we line up with the A, we line up with the C, here we go. CAC codes for histidine. And so that's how it works across life. But let's go back to the final one. And so this is just the basics here, but you should, if you see a DNA strand, you should be able to tell me what the RNA strand is, and you should be familiar with what the start codons are and the stop codons. Some topics just need more explanation, and that's why I'm here. So reach out. Send an email to kbyers@mineralarea.edu. I can help give study tips, we can set up a phone call, or even a video conference.